Okay, I think we might start. I'd like to welcome everyone to this Adelaide Biomed City ABMC Research Mini-Reviews. And we've got in excess of 80 people already logged in, so that's fantastic. And I really appreciate everyone logging in today for this session, which has a theme of genomics. Before we start, I'd like to acknowledge that we're on Kaurna land and acknowledge elders past and present, and also acknowledge any Aboriginal or Torres Strait Islanders on the call today. So today we have two speakers, and I also just wanted to emphasize that if people want to ask questions, they go to the Q&A slot at the bottom of their screen and they can type in their questions. I'll then paraphrase their questions to the speaker. We now move on to our second speaker, which is Paul Thomas. And Paul is also part of precision medicine at SAHMRI, but it is, and I want to point it out because of the precinct, that Paul's a joint appointment between us and the University of Adelaide. So terrific collaboration across the precinct. Paul's the director of the genome editing program of the precision medicine theme, and obviously a professor at Adelaide University. And Paul will be talking to us about the development of preclinical models and novel therapeutics using CRISPR genome editing technology. Go, Paul.
Great, thanks, Steve. I'm just going to share my screen. Hopefully you can all see that. Just let me know, Steve, if it's not coming through. Great, okay, so yeah, thanks very much for the opportunity to speak in this really, I think, interesting and useful forum as part of the ABMC initiative. So we are interested essentially in two things. Our research focuses, and has for many years, on disease modelling in mice primarily and also in cells. We're interested mainly in monogenic diseases, and one example that I'll give you today is a form of epilepsy that we've been working on for a number of years. And what's made this, I think, much more, it's facilitated this research aim that we have to understand the pathology that underpins these diseases, is that we have access now to a genome editing tool, CRISPR-Cas9, which I'm sure is familiar to many of you in the audience, so that we can actually move quite rapidly into the modelling phase and then get to the more interesting part, which is understanding how these diseases are caused. The other research aim that we have is to also use CRISPR-Cas9, and this is a more recent aim for my group, and it's one that's really focused on correcting these disease-causing variants that occur in a number of different diseases. We're focusing again on monogenic diseases for obvious reasons, and trying to pick ones where we feel that we've got the best chance of correcting the disease-causing mutation in the shortest amount of time. And I'll tell you about two examples of that as we get through the talk. So CRISPR-Cas9 really, as I said, needs no introduction. It's a form of molecular scissors. Many of you will have used it in your own groups, I'm sure. I just wanted to point out a couple of features of it. The first one is that it can be used as a pair of scissors basically to break genes or to create specific deletions in genes simply by adding it into the cell. And in that context, you don't need any form of repair template, but you can essentially knock out a gene or delete an exon by using a pair of CRISPR scissors, and that's a very efficient process. It can be also used to insert specific sequences at a site within the genome, but to do that, you need to add a repair template. And this is a much less efficient process than the simple deletion. So the take-home message here really is if we want to use this in the context of therapy, the best therapeutic targets or the best disease targets are going to be ones in which we can avoid the use of the repair template and simply add the CRISPR scissors to restore function. And I'll get onto that in a moment. First, though, I want to tell you a little bit about just a brief little vignette around a gene that we've been working on for a long time, Protein 19, originally and still with Joseph Gex's group, and he was responsible for finding this disease-causing gene. It's a form of epilepsy, but the unique feature of this, the really unusual feature, is that it's an X-linked gene and carrier females, so heterozygous females, are affected, whereas they're hemizygous males, so they only have one X chromosome, of course, and this contains the disease variant. They're unaffected. So there's been a lot of conjecture around what's the mechanism that underpins this disease. So we use CRISPR-Cas9 in mouse models to get to the bottom of this and published this quite recently. You're welcome to look up this paper. I'm happy to discuss it, of course. Basically, what we did is we created a mouse using CRISPR-Cas9, which had a little tag, an epitope tag at the end of the wild type sequence. And this is an example of a control brain from an embryo that carries this tag in the presence of a wild type chromosome on the other X chromosome. So this is the wild type control. And the pattern you see here in green is the arrangement of the cells in a wild type background. However, when we create the disease model shown here on the right-hand side, you can now see that there's a striking segregation of the wild type cells shown here in green versus the knockout cells, which are shown here in this pink color. And I guess this is just an example of how we can quickly get to a disease mechanism using CRISPR-Cas9 technology and make quite an inroad into the cellular arrangement, the disease phenotype in terms of the cellular mechanism early during brain development in this particular disease model. And we know this is having an impact on the brain activity in these mice because when we use ECOG to look at their activity, we can see there's quite an elevated pattern, as you might expect for an epilepsy model that's not present in the controls. And now we're quite focused on trying to connect these two phenotypes using CRISPR technology to work out exactly how the cells are connecting with each other at the level of the synapse. Unfortunately, there's not time to go into that in more detail. The next example I want to tell you about from our research group is one that's more focused on this therapeutic aim, in which we're tackling a disease which is not uncommon, Duchenne muscular dystrophy. Again, it's an X-linked condition. And this is a little bit of an unusual condition in that the gene that causes it, the DMD gene, is huge and it's prone to exon deletions. And the most severe form of the diseases occur when, for example, an exon is deleted that causes a change in the open reading frame. And that's indicated by the different ends of these two exons you can see here. So an exon 51 deletion changes the open reading frame. This, of course, causes a truncation of the protein, and this results in a severe disease. The interesting thing about DMD is that if you have a second mutation, or if you have a mutation which in fact removes exon 51 and 52, this doesn't change the frame as shown by the ends here of the exon, and this results in a much milder disease. And we believe this is a good target, as do many others, for CRISPR therapy, because we can imagine correcting a disease-causing mutation with an exon 51 deletion by, for example, removing the next exon in the chain, thereby restoring the frame of the protein. So this protein reframing approach is of interest, as I said, to many. And again, we like it because it doesn't involve adding a repair template, which, as I said, is a much more efficient way to correct a disease-causing gene. So this is just a snapshot of some of the data. I don't have time, obviously, to go into the detail, but what essentially you're looking at here is this band here is the exon deleted product, and the top band here is the full-length transcript, and we've just amplified the relevant region. And what we tried here are all sorts of different guide-based strategies to remove the exon and see if we can get exon skipping. And as you can see, there's a couple of examples here of some of our strategies that we have generated in the lab, quite efficient, up to 95% exon skipping. So we believe this has quite a lot of potential moving forward towards a therapeutic phase. Obviously, there's a lot of work to do in this area to try and validate these kinds of approaches. We're using mouse models for this, and as I said earlier, it's quite efficient to generate mouse models in which you can remove an entire exon by adding 2 CRISPR guides. So this is one that we've already made. We've made the exon 51 mouse model. We're in the process of also building humanized mouse models as part of our strategy to actually look at several different exons that cause, when deleted, that cause DMD. So quite a lot of work in this area in the group, and I obviously don't have time to get into it in any detail, but I'm just acknowledging the group members as we go through the talk at the bottom of the slides. A second disease that we are just beginning to tackle now, which I don't have time to go into again, but is one that I think could also constitute a good target for CRISPR therapy, is ADRP. And the reason for this is, again, we can use this strategy whereby we're looking to knock out a gene rather than adding a repair template to restore it back to a wild type state. And what I mean by that, in this particular case, this is an autosomal dominant toxic gain of function or dominant negative mutation. This particular row mutation is the most common one. And what we're attempting to do here as our strategy is to use a guide RNA that's a perfect match for the mutant allele, which should target it, therefore, for inactivation, but shouldn't touch the wild type allele by virtue of the fact that the wild type allele contains one base pair that's different to the mutant allele. And it is possible to get this degree of specificity with the CRISPR system. There are some caveats to that, but already we have some good candidates for this approach moving forward. We also have a little bit of fun with other aspects of CRISPR that there's not a lot of time to mention, but I just wanted to touch on them as we go through. We've been able to use CRISPR to delete entire chromosomes from cells, the X chromosome, the Y chromosome, which was published a couple of years ago. Adi Kazuma, published down, publication is here, was the first author on a study which showed that in fact CRISPR-Cas9 generates in some cases large deletions at the single cut site, which has importance, or it was not recognized at the time and has implications for therapy. And we also play a little bit with other CRISPR systems that are published and use them to generate mice and cell lines with greater efficiency. We also interested in CRISPR gene drives. It's not a biomedical application really primarily, but it's one in which we can hope to modify an entire population of mice in the wild by virtue of the fact that the CRISPR gene drive replicates itself in the germline of animals. That's been a project that's been going for some time in the group, and we have several group members working on that and different strategies. And the last thing that I just wanted to mention before I finish is that a number of years ago, really around 2014, 2015, I established the South Australian Genome Editing Facility, or SAGE, and this was with support initially from University of Adelaide, and then through SAHMRI, and now through Genomics Australia. We provide a subsidised one-stop shop for mouse generation, and we've generated a lot of lines over the years for clients, both within this precinct, but also externally. And my take-home message here is if you're interested in having a mouse for your own research projects, don't hesitate to let us know, as this is a subsidised service. And the email address is on the bottom right. So with that, I'll conclude. Thanks again to SAHMRI for hosting the laboratory and great collaborations here. And last but not least, thanks to my group who make it all happen. Thanks, Dee.
Thanks, Paul. What a terrific talk. Fantastic. I might start off the questions, Paul. When you're thinking about utilising this in a medical therapy, and I was thinking about the eye example, is the eye a good example where you could perhaps, do something localised within the globe rather than having systemic impacts of the CRISPR therapy? Yep. Is that something that people have already done?
Yep, we haven't worked on eye before, but there has been a bit of research in this area, and that's one of the reasons we've chosen it. It's possible to introduce AAV vectors into the eye. There's some degree of immune privilege in the eye, and of course it's an accessible organ. So one of the difficulties about tackling internal organs with this CRISPR therapy type approach is access to the region of interest. Here, I think the eye should be a good target for us to test out some of these strategies.
Thanks, Paul. There's a question there. I think you can probably see it from Michael Ricos. Have you seen brain malformations for the PCDH19 mutant mice as per human patients? And have you made mosaic male mice mutants for PCDH19?
Michael, hello, Michael.
Females. Sounds like a good question for you, Paul.
Yes, we haven't seen overt brain malformations as we see occasionally in humans. That's a difficult question because each female brain is specific in terms of the X inactivation pattern. So essentially you're dealing with a unique mouse in each case, as is the case in humans. Each female is unique. We did find a small percentage of females that had brain malformations, as Michael alludes to. We haven't seen anything that reminiscent of that in mice, but of course there are also differences in terms of the way the brain forms in mice and humans. We did speculate that a ferret might be a good model for that because that has it's accessible and has a more similar brain structure than mice do. And the other question was around mosaic males. We tried to generate mosaic males, but it didn't work in the first experiment. We tried to do that by injecting the CRISPR into, say, 4-cell embryos and therefore mutating some of the cells, but leaving some of the cells wild type. It didn't work in the first experiment, and we haven't been back to that one, Michael.
All right, thanks, Paul. Perhaps one last question. The mouse population studies, where were you going with that? What's the goal of those studies?
Primarily, this is driven by conservation goals, particularly on islands where mice, once rodents get onto an island, it's a disaster for the local bird population because they eat the chicks, they eat the eggs, and there's been a lot of extinctions on islands. So currently that's addressed by dumping bait on islands, which is expensive, it's toxic, it's not humane, and doesn't always work. So the idea of this genetic technology is to seed the island with a so-called gene drive, which would spread throughout the island and ultimately create conditions for a genetic modification, which would either, for example, reduce the population by female infertility or convert all the population, for example, to males, which would also cause a crash because females would then become limiting. So both of those strategies are being actively pursued by us and a few other labs. And it's pretty early days on the CRISPR gene drive approach. But I would say in insects, it's progressed quite well with the objective of malaria control, either getting rid of the mosquitoes completely from the population or passing a malarial resistance gene through the population using gene drive.
Right. And we are over 5 o'clock, but I'll get this last question in. And that is, do you see the mouse population studies branch out to other species? And I think you mentioned ferrets, but are there other species you're working on?
Yeah, no, ferrets was that sort of lowest.
The large animal facility out at Pearl is very excited. Possibilities.
Probably, if it works in mice, the next cab off the rank would be rats, because they are probably the most damaging pest, small rodent pest. There is interest in cats and other species, but this is, yes, it could be applied to other species, but I think mice are the best target because we can manipulate the genome, and we have access to them as a laboratory target species. They're also a lot cheaper than any other species.
Okay, all right, we're at 5:03. We've still got 106 participants, so that's brilliant. And we've got some thank yous on the Q&A, which which I'm sure the speakers appreciate and I as chair appreciate. So thank you for the thank yous. And I'd just like to thank David and Paul for brilliant presentations. Short, sharp, to the point, really very good. And I think this is going to be an excellent thing, the ABMC Research Minireviews, and I encourage everyone to log on next week for the next one. So thanks, Paul and David. Thanks everyone for logging on, and have a great evening. See you later.